So we will be teaching today what are the abnormal constituents present in urine. First of all, you will have a rough idea what are the constituents present. Then I will be teaching in detail about how to do the Benedict test for identifying the reducing sugar and the non-reducing sugar, and respectively in the later classes rest of the abnormal constituents like protein, ketone bodies, as you have said. So these things will be taught by other faculties one after another. So there are some tests that I will show over here, the names of the test for each and particular constituents, but all of the tests we are not going to do. Some of the tests will be only theoretical, you have to know, because this can be asked in your viva. Okay, the names, the procedure and the inference that what is going to happen, and some tests which are very important that we will be showing you, will be demonstrating you, and you have to perform on your own self. Once I am done with this lecture class, I will be sending you to that lab. You people have to separate yourself in 4 different groups. Okay, separate groups among yourself, make as you are feeling comfortable, and after that you will have to do the test on the Benedict test that I will be discussing on with you. Fine? Okay, let's start the class. Introduction. There are many pathological constituents occur in traces in normal urine and escape the detection due to low sensitivity. Now, can anyone tell me what is sensitivity? Here it is written that it's a low sensitivity. Sensitivity means test is low sensitive. Do you have any idea? I'm just trying to give you a basic idea about what the terminologies are. Because if you don't understand the terminologies, you will not understand. Sensitivity means you will do the test. The test might show positive, might show negative. Okay, if the quantity is less in urine sample, if you are doing the test, it might show positive, it might show negative. Okay, so we cannot say that the urine sample don't have sugar. Let's say the sugar is very less. You are doing Benedict test. The test is showing negative. Now you cannot say that the sample doesn't have sugar. It is having sugar, but the amount is so less that the test cannot detect it. That is a low sensitivity. That is why whenever we are doing this kind of test, the amount should be more. If the abnormal constituents are more, then only these tests can detect it. Fine. Now, all the methods, then their concentration are increased markedly in the urine in different pathological conditions. I will be discussing what are the diseases, in which disease, which condition, which elements increases. For an example, the reducing sugar will increase in diabetes mellitus. Fine. Now, galactosemia, glycosuria. These are some names. I will be explaining one by one. So these are conditions where the reducing sugar will increase. Ketone bodies will increase in diabetes. The complication of diabetes, if you are uncontrolled diabetes, you are not taking any medication for diabetes, then you can land up in diabetic ketoacidosis, where the body metabolizes the ketone bodies and the ketone bodies comes out in the urine. Okay, so that is an example of ketone bodies getting in the urine. Now you have some kidney pathologies. The kidney is not functioning, the kidney is not filtering properly. Then what happens? The proteins which is there in the body, which should have been reabsorbed by the kidney, that is coming out in the urine. So these are some basic examples. What are the abnormal constituents and how it can come in the urine. Fine. Now, on standing, urine undergoes bacterial fermentation. We all know that urine is, if you keep the urine sample over in a table, then obviously with time the organisms will start growing over there. Fine. On standing, urine undergoes bacterial fermentation. Now, it can be preserved under refrigeration or by adding chemicals like toluene or chloroform. Now, you can preserve the urine sample because we are not testing the urine sample immediately. Let's say you will collect the sample over here and the test will be done somewhere else. Now, you cannot send it just like that. Otherwise, the urine will grow organisms. And whenever that person will do the test, they will see some microorganisms are there. So, they might be thinking like probably it got infected. The patient is having UTI, urinary tract infection. Because of that, the organisms are there. But the case is otherwise. The organism grew because it was not preserved properly. So with what thing we can preserve? That is the toluene and chloroform. This one. Fine. Now, usually the analysis is carried out with properly preserved 24 hours urine specimen. So that is the gold standard. Gold standard means this is what we should do, but this is not possible. What is 24-hour urine specimen? That is, you have to collect the urine sample from the patient from 8 a.m. till next day 8 a.m. That is 24 hours urine sample. Then you have to take the sample from there, then you have to do the test. But we don't do it. That is why we take the early morning sample, the fresh voided urine. Fine? That is the sample we take. Next. Now, physical characteristics and pathological conditions. Physical characteristics, it has been already covered by ma'am. You all know what is the appearance. How will it appear? It should be clear. But in pathology, what will happen? It will become turbid because of the constituents present in the urine sample. Like protein is present, sugar is present. This will make the urine turbid. It won't be clear. It won't be clear like a water. Fine. Next comes the odor. What is the normal odor? The normal odor is aromatic in case of urine. The odor of normal urine is aromatic. Ammoniacal, it becomes in long standing. When you keep the urine for a longer period of time, it gives you the smell of ammonia. Why? Because it has urea, ammonia both. That's all. Now, some pathological conditions where the odor can be very specific. Like in case of MSUD. What is the full form of MSUD? Madam told. Anybody can recall what is the full form of MSUD? Maple syrup urine disease. Fine, remember it, maple syrup urine disease. In that case, you will get the smell of like a burnt sugar. Okay, now there is another example, ketone bodies. In case of ketone bodies, which kind of smell you will get? That is a sweet smell. Fine, there are some more examples like phenylketonuria, PKU, in short the terminology is phenylketonuria. In that case, what will be the smell? The smell will be like mousy odor. So there are many more. These are just few examples. Okay, there are cabbage-like smell. So particularly to disease it varies. These are some examples that you have to. These are must know. Fine. Next, the volume. The urine output is increased in conditions like diabetes or effect of drugs like diuretics. Anybody know what is diuretics? Frequent urination. Yeah. There are some patients who is suffering from renal diseases. They cannot void, they cannot pass urine. Okay, because the kidney is not functioning properly. So initially what we do, we give them diuretics. Diuretics does what? It forcefully takes out the water from kidney. It makes the kidney work and take out the water from there. That is diuretics. Okay, so if you are giving someone diuretics, somebody who is voiding, or some patient is coming with puffy face, swelled up leg because of kidney pathologies. Now you have to take out the fluid from their body. How will you give? You give the diuretics, they will void more, they will urinate more. So that will lead into polyuria. That is drug induced because of diuretics. Fine. And some people generally they are having diabetes, rather be it diabetes insipidus, rather be it diabetes mellitus, doesn't matter, they will land up in polyuria. Diabetes mellitus, this is because of some other pathological condition, and diabetes insipidus because of some other reason. The etiopathogenesis you will learn in pathology, that is fine, but in both the conditions there will be polyuria. Same, there can be oliguria, there can be anuria as well. Now you people know, you are acquainted with the terminologies, right? Oliguria is urine volume how much? Oliguria, not anuria. Fine. Oliguria is the urine volume should be less than 500 ml. Fine. And anuria means complete cessation of urine, where the volume will be less than 100 ml. Fine. I'll be asking questions, don't be like mum and just be silent over there. I'll be asking questions to every one of you in my next class. Now there are some examples where can be anuria. Okay, that is the complete cessation of urine. First is shock. What is shock? Anybody has any idea what is shock? Shock, not the electric shock. S-H-O-C-K, shock. There are different types of shock that we'll learn in pathology, but for now you have to know what is shock actually. Let's say patient comes with infection, severe infection that we term it as sepsis. Okay, the patient will have hypovolemia. That is, patient blood pressure will be less. Okay, hypotension, and patient will end up with comorbidities, other associated comorbidities. Now when the patient is having low BP and pulse rate is high, tachycardia, these are some, and patient's GCS, that is a consciousness. For now I can say is like patient consciousness, whether the patient is conscious or comatose, whether the patient is semi-conscious, unconscious, depending upon that we say the patient is having shock or not. In shock what happens, the multiple organ gets involved because of the infection, and then when it involves the kidney cannot function properly, and that time your urine output also decreases. That is the representation of shock. Fine. Now in incompatible blood transfusion, you are well acquainted with physio, right? Incompatible blood transfusion, if the ABO is not matching or the RH is not matching, in that case there will be hemolysis. Fine. And that can lead into complete cessation of urine. Fine. Mercury poisoning, acute nephritis, these are certain examples of the disease. Now color, urine becomes smoky brown in presence of blood. Fine. If there is blood, it will become smoky brown. If there is bilirubin, it will become yellow. Now can anybody tell me why urine color is pale yellow? The normal urine color is pale yellow. There is no bilirubin. Why it is urochrome? Nice, very good. Now black when the melanin is present. Now can anyone tell me what is melanin and what is its work? Exactly, skin pigmentation. This gives our skin the color. If somebody is having less amount of melanin, they will have fair skin, right? Like foreigners, Europeans, they have less pigment. And this melanin is very good for our skin. People tend to say like you are dark, but the darker skin people are much more well equipped to prevent cancer. Exactly. If the melanin is more, there is less likely to get a cancer. But the fair skin people get more easily, there is chances of getting cancers. Fine. Now there is one more condition, that is the alkaptonuria. Here it is written alkaptonuria. Why it is important and what happens in the sample? Let's say you take a urine sample from a patient and you keep it over there. As soon as you keep it, it will turn into black with time, without any reaction. When you took it, it was normal color. But when you are preserving it or just keeping it somewhere, it will turn black. Why? Because of the presence of homogenesic acid. Fine, HGA, homogenesic acid. Because of that, this becomes black on standing. That is seen in case of alkaptonuria. This all can be asked to you as a clinical question, like a patient comes with an urine sample. Whenever he was voiding, the urine sample was normal in color, but after he finished urine, he saw that the pan is black in color, dark in color. Why? What is the clinical importance? What diagnosis you can come to, and what is the causative agent because of which this black color is coming in the urine? This can be asked in your vivas. Fine. Now urine becomes milky if pus, bacteria, epithelial cells and lipids are present. I don't think I have to explain this part, because if there is milk present, if there is pus present, what is pus? Ma'am told? Yes, dead white blood cells, not the red one. Okay, WBC. And lipids are present, this will give the milky white in color. Fine. Now pH. Urine is significantly acidic in fever and diabetic ketoacidosis that I was talking about, when ketone bodies will be present in the urine sample. Fine. Decay. And the specific gravity, it is increased in acute nephritis and decreased in diabetes insipidus. Now can anybody tell me why in acute nephritis the specific gravity will increase and it will decrease in diabetes insipidus? What do you understand by specific gravity? Anybody has any query about specific gravity? Why you will get that last point? Why you will get increased specific gravity in case of acute nephritis and decreased in case of diabetes insipidus? Any question came to your mind why it will happen? What is diabetes insipidus? When there will be more and more amount of urine. Urine will become more thinner. The constituents will be less in urine. The constituents are normal. The constituents are normal. Constituents are fixed. But the urine volume is so much that the density becomes less. When the density becomes less, the specific gravity will also decrease. But in case of acute nephritis, why is it high? Concentrated. Because? No, the water content is there. In case of acute nephritis also, the water content will be there. Pardon? No, so try to say.
Water reabsorption is.
No. In case of acute nephritis, what happens? The term nephritis means what? Kidney inflammation. Itis means inflammation. Nephro means kidney. Kidney inflammation. Any kidney pathology, what will happen in there? The kidney will not be able to reabsorb the particles. So what will happen? The particles will come up in urine. If the particles are coming in urine, the urine amount is fine. In diabetes insipidus, the volume is increasing. But in case of acute nephritis, the volume is fixed, but the constituents are more. Because the constituents are more, the specific gravity will increase. Fine? Next. Now these are the list of the abnormal constituents that I was talking. What you can get? The reducing sugar, protein. There is one called Benz-Jones protein. It's a specific protein that can be found in case of multiple myeloma patient. One line can be asked from here, that is, in case of Benz-Jones proteins, in which clinical condition you will get the Benz-Jones proteins in case of urine. Fine? That is multiple myeloma. I repeat, it is multiple myeloma. Okay? Very repeatedly asked question in NEET PG exam. When you will finish your final exam, these are the questions that is being asked. And when you are going for Viva, examiner will ask you basic questions at first. If you can say everything, they will ask you this kind of questions to give you more marks. That is the honors marks that we say. Fine? Then blood can be there, ketone bodies can be there, bile salts and bile pigments can be there. Fine? Next. Now this is my topic proper for today. Test for the reducing sugar. What is the test name? Benedict test. Fine? And these are the colors. And test for proteins are here. Heat coagulation test, this is what we are going to do. The heat coagulation test we will do, and rest of the two tests you have to know the names and the detail. That is the sulfosalicylic acid test and the Halon nitric acid test. This will be done by other tutors in the later on classes. Today I will be concentrating on this. Fine. There are some more slides I have just collected for you people. This is the Benz-Jones proteins test, the flocculation and the Osgood-Haskin test, and this is how it will look like, the flocculation test. Fine. And this is for the blood, that is a benzidine test. When you will go in the topic proper, you will get to know in detail what we are going to in the test. Okay. Next, this is for the ketone bodies, the Rotheras test and the Gerhardt test. We are going to do the Rotheras test, and this is the example of the Rotheras test. This is the purple color ring that will appear in case of Rotheras test. Fine. And then comes the bile salt test. For bile salt, this is the Hay sulfur test. Very easy test. You have to take a sample and you have to take a constant. That is the distilled water, and you have to put the sample in it, and after that if it sinks, the Hay sulfur test is positive, otherwise it is negative. That's all. Pardon? The enediol, which is a very strong reducing agent. Okay, fine, that is written over here. See, in hot alkaline medium, the reducing sugar gives enediol, which is a strong reducing agent, which reduces the cupric ions to cuprous ions. Now, if I tell you the reagents, when I will be telling you the reagents, the constituents of the reagents, then you will be understanding from where these cupric ions are coming from and how these are getting converted to cuprous ions. Fine. And forms the cuprous hydroxide, which then gets converted into the cuprous oxide, that is the Cu2O, and it forms the red precipitate. When you are doing the test, if the glucose content is more, stop taking photos, you will be circulated with the PPT only. You can take the PPT. Concentrate on the class. In Benedict's, what I was talking, the cupric ions get converted to cuprous ions and forms the cuprous hydroxide, which then gets converted into the cuprous oxide, which forms the red precipitate. Somebody asked me in last class like why this red precipitate forms. Okay, so this is the reason, this cuprous oxide is the reason which is forming the red precipitate. And we don't judge the color of the test only by the red precipitate. As soon as you finish the test, after you boil, you keep the test tube for 5 minutes to 10 minutes for cool down. Once it cool downs, then you see the color and then you come to the conclusion of the test. Fine. In Benedict's qualitative test, the blue color changes into different colors depending upon the concentration of the reducing agents. This test helps in distinguishing between the reducing and the non-reducing sugars. So what it does, separates between the reducing and the non-reducing sugars. Fine. And what will be the color changes depending upon the concentration of the sugar. Let's say the sugar concentration is very less, then it will change into only green color. If it is more, then it will become yellow color. If it is more than that, then it will become orange to red color. And highest, that is the more than 2 gram%, then it will become red precipitate, that is the brick red in color. Okay, that is what is written over here in Benedict's qualitative test. The blue color of the reagent will change into different colors depending upon the concentration of the reducing agents. Next, now the composition of the Benedict's qualitative reagent. What I was talking, that from where this cuprous and cupric ions are coming. First of all, this is the copper sulfate which breaks down. When copper sulfate breaks down, what it forms? Copper and sulfate. Fine. This copper will give you the cupric ions in the solution. Right? Now this cupric ions will do what? This cupric ion will immediately try to react. Immediately try to react with the agents, whatever is there in the solution, and give the precipitate like that. But if it does it, then the test will not take place. So we have to keep the continuous supply of copper. And with what we will do that? That is the sodium citrate. It prevents the precipitation of the cupric ions and forms a loosely bound complex with the cupric ions and forms the sodium citrate cupric complex. This is the complex that is forming. So basically what is happening, sodium citrate is there in the reagent. Sodium citrate is reacting with the cupric ions, forms a loose bond between them. It's not strong that it is not going to break only. It is loose bond. It will break eventually, but for the time being it is making a loose bond so that on an instant the test doesn't take place. The test takes place over time. That's why we heat it for some time, and till the time it is heating, the reaction is taking places in the alkaline medium. Once the reaction takes places, the reaction keeps on going. That's why we have to keep the test tube for 5 to 10 minutes to cool down. Once it cools down, then only we will come to the conclusion what is the color of the sample. Fine? Understanding? Anybody has any difficulties in understanding? Can raise hand if anybody has any difficulties. Okay, now the third point is sodium carbonate. What it does? It makes the medium alkaline. Now I told you the medium have to be basic, the medium have to be alkaline. So for that alkaline medium we are using the sodium carbonate. So these are the 3 constituents of the Benedict's reagent, which is very, very, very important. You all have to know the names and the function. What is the name of the constituents and the function of the constituents. Fine. Now the procedure is very simple. It's a very simple test. You have to take 5 ml of Benedict's qualitative reagent in a test tube and add 8 drops of urine and mix it well. Fine. Then you do what? Boil the solution for 2 to 3 minutes and wait for the color change. Now as soon as the color change occurs, you keep the test tube in a rack and wait for time to cool down. Once it cools down, you see a change in the blue to different colors like green, yellow, orange, red and brick red that I already told, depending upon the concentration. Can anybody recall and tell me the concentrations from last class who have attended? You say? 0.5 to 1 for? Are you sure? Less than 0.5. Less than 0.5 is green. After that, 0.5 to 1. That is yellow color. And 1 to 1.5? Orange to red. No. My bad. The 0.5 to 1. It was 0.5 to 1, then 1 to 1.5, 1.5 to 2 and more than 2. Those were the concentrations. Now, somebody may ask, in last class also someone asked me like, why 8 drops of urine? Okay, why not more than 8 drops? Why we cannot use more than 8 drops of urine sample during the test? We can use 6 drops as well, we can use 10 drops as well. Why 8 drops only? Okay, so this is the reason, because in 8 drops in urine, because if you are using higher concentration of urine, there are some byproducts in urine which is also there. Urine is not only filled up with sugar, proteins and ketone bodies. There are many byproducts are there. Those are metabolic byproducts. For example, this ascorbic acid, which is also known as the vitamin C. Okay, so ascorbic acid, glutathione, homogentisic acid, these are some constituents which is also present in urine. Now if you are using 10 drops of urine, these constituents are also high in comparison. So this test will give also the positive test result. Whereas the sample does not contain sugar only. But because of these byproducts, the test will show positive. So you will come to a false conclusion like the patient is having reducing sugar in their urine sample. Whereas the patient is not having. It's your mistake that you added more than 8 drops. That is why we don't add more than 8 drops in our test. We add only 8 drops of urine sample. Fine. Now this is the reactions that is taking place, that the reducing sugar reacting with the sodium bicarbonate and forming NADOLs. Sodium bicarbonate because it is alkaline. Forming NADOLs. NADOLs is the strong reducing agent which will reduce the cupric ion to cuprous ion. Fine. Now the cupric ions are getting converted to cuprous ion, and cuprous ion later on binds with hydrogen and water and results in cuprous hydroxide, and later on it forms cuprous oxide, Cu2O, and which is forming the brick red color precipitate. Fine. Now this is the interpretation that you will get. The blue one, when this is only the sample, there is no reducing sugar, the color remains as it is of the sample. Then if there is traceable means 0.5 to 1%. Here it is written only 0.5%, but you can say like it is 0.5 to 1%. It will be green. Now more than 1%, 1 to 1.5%, it will be yellow. If it is 1.5 to 2%, it will be orange red. Fine. And more than 2%, that is brick red. If somebody gets brick red, that is definitely either they are having some of the pathologies. It can be either diabetes mellitus, in which the sugar is high. Why? Because the insulin is not working in our body. Second reason, the kidney is not working properly. Your body sugar is fine, but the kidney is not being able to take up the sugar from the sample, and it is getting excreted in the urine in higher concentration. That can also give you a positive test. Another example, let's say you are taking 100 chocolates today. So sugar content is too much in your body right now. So you can also get not the brick red, but some amount of sugar can be present in your urine sample, which is not a disease, but it is because of the diet. That is how diet also affects the urine color and urine constituents as well. Not always it have to be disease only. Now, some clinical interpretation. A very small amount of reducing sugar may be present in urine, and this is insufficient to give the positive result. That I was saying in the first line, that if the constituents are less, it might give you a negative result because the test sensitivity is low. We are doing it as a bedside test, that is just to know the patient is having reducing sugar in the urine or not. That's all. That is why the test sensitivity is also less. There is two terminologies. One is sensitivity, another one is specificity. Specificity means this test is very specific. Fine, very specific. Like in case of ketone bodies, we are doing the Rotheras test. If the Rotheras test is positive, that means it contains ketone body. But if it is not positive, it doesn't mean that it is not having ketone bodies. There can be ketone bodies. The sensitivity is less, but the specificity is very high. So these terminologies are a little bit difficult for you, so just trying to break it down the terminologies if you understand. Any difficulties in understanding, please raise your hand so that I can explain again. Later on, it should not be like, I don't know what is sensitivity, I don't know what is specificity. Just to get acquainted with the terminologies. Fine. Now, in any common laboratory. Now, significantly increased amounts are observed in several conditions. Like some of the important ones, as I have already said, diabetes mellitus is one of the conditions. Then lactosuria. When you can see lactosuria? What is lacto? You break the word, you will understand. Lacto means milk. When can be found? Either in a neonate who is already on a breastfed, or else a mother. Why mother? Because in mothers there is a hormone called prolactin which is high. That is helping you to create the milk, and oxytocin is ejecting the milk. Fine. So because of that, in pregnant mother who is feeding the baby, they can also have lactosuria, which is normal, which is not a pathology, it is not a disease. Fine. But they can have lactosuria, and in that case also the test will become positive. In renal glycosuria, that I was saying, like the pathology is there in your kidney. Your blood sugar level is normal. The blood sugar level that I have, the patient have, same. But the patient's kidney is not working. Because of that, the patient's kidney is not being able to reabsorb the glucose from the blood. And that's why it is coming out in the urine. Fine. That is renal glycosuria. And then comes the galactosuria. Now, galactosuria is a condition. This is a topic which is being asked again and again in NEET PG exam as well as in Viva. If you are doing very good in Viva, they can ask you about this galactosuria. What is galactosuria? First of all, let us know what is galactosuria. Galactosuria is a genetic condition, inherited condition. The defect in the gene is the GALT gene. Okay, the GALT gene is defective in both the mother as well as the father. Because of that, the baby is also not having the GALT gene. Baby's gene is also defective, and because of that, the baby is getting the galactosuria. That is the galactose, like glucose, fructose, sucrose. The galactose component is coming out in the urine. Fine. And what is the enzyme that is responsible? That is the GAL1 put. Full form is galactose 1-phosphoruridyl transferase. Fine. At least remember the short name. I'll write down the full name in board. You can copy it later on. GAL1 put, that is the enzyme which is defective, and GALT is the gene which is defective. So be conscious what is being asked. If the gene, then the GALT gene. If they are asking the enzyme which is responsible for this condition, then it's a GAL1 PUT. That is galactose 1-phosphoruridyl transferase. Don't talk in my class. I'll throw you out of the class. Now these are some extra ages. Extra ages means these are for your knowledge. Later on it can help you in your NEET PG exam, and if somebody is getting same very good in Viva, these are some exceptions per se. Fine. Now some non-sugar reducing substances may also give false positive results, like creatinine, ascorbic acid, urates and glucuronides. These are non-reducing sugar, but still they can give positive result. So this is the exception to the reducing sugar. Normally what we are doing is Benedict test, we are detecting the reducing sugar is present or not. Now these are some exceptions which are non-reducing sugar. This will also give the positive test. Okay, so exception might be asked, like which are the non-reducing sugar which shows Benedict test positive. Fine. And some drugs are also there that we use, that like salicylates. PAS is the para-amino salicylic acid, and isoniazid. Now isoniazid is one drug which is widely used in TB treatment. If the patient is having TB, we use isoniazid. That can also give false positive results. Fine. Reducing sugars like sucrose, starch and other polysaccharides gives negative test. Why sucrose gives negative test? Anybody has any queries regarding it? Pardon? No, it is not a non-reducing sugar. Sucrose is not a non-reducing sugar. Other starch is a non-reducing sugar. Polysaccharides are non-reducing, but sucrose is not. Sucrose is reducing. But sucrose is a disaccharide. It contains what? Glucose and fructose. Exactly. Because of that reason, it is not showing the test positive. The Benedict test mainly happens in monosaccharides. Always remember, it mainly happens in the monosaccharides. So sucrose is a disaccharide which contains glucose and fructose both. And because of that, it also don't show the Benedict test positive. Now, if reducing sugar is detected in urine, If reducing sugar is detected in the urine, blood sugar level must be correlated with the findings. Now, someone give me one example why this is important. You got reducing sugar in the urine sample. Now they are telling you to check it with the blood sugar level as well. Why it is important? I just gave you an example. Just now I told renal glycosuria. What happens in renal glycosuria? The blood level of sugar is normal, but the urine glucose is high. So if you are cross-matching, you will understand that the patient is having renal glycosuria. Otherwise, you will be like the patient is having diabetes mellitus only, because the reducing sugar is high in urine, and you are coming to the conclusion the patient is having diabetes mellitus. No, the patient is not having diabetes mellitus. You have to cross-check with the blood sugar level. If it is also increased, then DM. And if it is not increased, then it is renal glycosuria. Fine. Nowadays, presence of glucose in urine can be confirmed by dry chemistry. These are advanced technologies. We are not going to do it in our lab, but just for your knowledge, with a glucose stick, the person can check their urine sample at home only. You don't have to go to labs also. These are so advanced techniques. And some paper chromatography technique is also there to identify the specific nature of the sugar, if it is required to confirm the diagnosis. Now let's say you have diagnosed the reducing sugar is present. Now you have to understand which type of reducing sugar it is. It is glucose, it is fructose, it is maltose, you don't know. So that's why comes the paper chromatography that can help you understanding the specific type of the reducing sugar. Which type of specific reducing sugar is it? Fine. Now these are some question and answers. Let's solve it. What are the common causes of oliguria? You all know about oliguria, right? What is oliguria? Less urine. How much? Less than 500. Anuria is less than 100. Less than 500 ml in how much time? 24 hours. Over 24 hours. Fine. Now what are the causes of oliguria? Dehydration. Yes. Any other example? No, diarrhea. Yeah, with diarrhea only you will get dehydration. And any other example? Someone from the last? Yeah, chronic kidney disease. Yeah, there also you will have oliguria. Now, what are the common causes of polyuria? Insipidus and mellitus. What are the common causes of glycosuria? I said only glycosuria. It could be renal glycosuria as well. It can be diabetes mellitus as well. So basically, if the reducing sugar or the glucose is more in urine, that will give you glycosuria. The same things coming again and again. You just get acquainted with the words again and again, listening to it again and again, and that's how you will remember the things. Fine. Now, what are the causes of hematuria? UTI, yeah, it can happen, and blood is present in the RBCs are present. To be precise, RBCs are present in the urine sample. How come RBCs can be present in the urine sample? Yeah, there can be some minor pathologies, there can be some abrasions, there can be some lacerations inside the renal, from kidney, ureter, bladder and urethra. In the whole pathway, if there anywhere there is any inflammation or infection or stone disease, when the stone is coming from the kidney and getting out of the urethra, what is happening? It is getting rubbed against the mucosa, it is eroding the mucosa, and that can lead to through the RBCs in the urine sample. Fine. In case of cancer also, in case of bladder, TB as well. In all these conditions, you will get hematuria. Fine. Now, why 8 drops of urine sample used in Benedict's test? You say, stand up. Say, yeah, exactly. So, why DM patients develop polyphagia and polyuria? What is polyphagia and polyuria? Anybody has any idea? Eating. The excess, the urge of eating more, that is polyphagia. Polyuria, too much urination. And polydipsia, too much drink water. Too much thirst. Yes, exactly. Too much thirst. So why they develop polyphagia and polyuria? Okay. Why polyphagia and why polyuria? Kind of close, but the reason is not good. Exactly, that is what. That is good. The insulin secretion is less. The insulin is less effective. It can be insulin less effective or the insulin is coming less. Whatever the reason is, because of that the glucose is not getting metabolized. If the glucose is not getting metabolized, you are not getting energy. For energy, you will try to eat more and more and more. That is why you are getting polyphagia. And why polyuria? Why urine more? No, the ADH is in diabetes insipidus. ADH is responsible for diabetes insipidus. Why polyuria in case of diabetes mellitus? Whenever glucose is coming out in the urine, it is taking water as well along with it. That is why the urine is more. The frequency is more. Whenever sugar is coming out, it is taking water along with it. That's why. Fine. Now, which drug causes orange color urine? I told you about one drug, that is the isoniazid, that will give the false positive test, right? In case of TB patient. There is one more drug in TB patient we are using, that is the rifampicin. Fine. Remember the name. Rifampicin is a one drug that is used in TB that can give the orange color urine. Fine. And which disease gives the mousy smell of urine? Lovely. What is isosthenuria? First of all, understand the word isosthenuria. Iso means what? What do you mean by hypo and hyperosmolar? The density is more, hyperosmolar. The density is less, hyposmolar. What is iso? Equal, equivalent. What is the specific gravity that should be there in case of isosthenuria? The specific gravity should be 1.010. If the specific gravity is this, then only it will be isosthenuria. Not more than this, not less than this. It have to be this. And any condition, clinical condition, any disease where you will get this isosthenuria, chronic renal failure. That is one condition, chronic renal failure, CRF. Okay. In that case, you will get this isosthenuria. The specific gravity is fixed to 1.010. Okay. That is an exception. That's why I put that question in. And what will happen to specific gravity in hypo and hyperosmolar conditions? Anybody has any doubts about specific gravity from last class? For each 3 degree, how much change will be there? For 3 degree change?
15.
No. For 3 degree change, for 3 degree, what is the base room temperature should be?
20.
20 degree. Write it down. 20 degree. And what is the room temperature given? 38. Not 20 minus 38, 38 minus 20. That is the temperature difference. Fine? 18. It's fine. Now, for each 3 degree change in room temperature, for each 3 degree increase in room temperature, how much we have to add? 0.001. Fine. Write it down. For 3 degree, how much? Not here. Here it will be only 3.
Into
0.001. Fine. Now plus here it will be plus with a base value because it is getting added with the base value. Now the idea is clear?
Yeah.
For the other rest of the students, if you are not understanding, let's say the room temperature is 38 degree. Fine. Your room temperature should be 20 degree. That is the basic we are catching hold of, because in Kashmir you are not getting 20 degrees centigrade. In Kerala you might not get it. The temperature is more. So you cannot change the environmental temperature. So you have to come to a base temperature, that is you are holding as 20 degrees centigrade. Now the temperature difference is how much? 18 degree. Fine. For each 3 degree, how much we are adding? This much. This is a fixed amount that we are adding for each 3 degree. So for 18 degree, how much we are adding? 6 into 0.001, coming 0.006. And the base value that we got is 1.020. So if you add with 1.020 with this much, how much you are getting? 1.026. This is the corrected specific gravity. You might be asked in your exam with a question paper just like this with a math equation. Just because you came to medical field, your math is not gone. Nightmare is still there. You have to learn. Fine? Yeah. Go. Take your seats. Yeah. Okay. Now, coming to the topic. What will happen to the specific gravity in hypoosmolar and hyperosmolar condition? Anybody? Don't try to mug up, just understand the word. See, medical field, it will be much more easier if you just breaking the words and understanding the meaning of the word. Once you understand the meaning of the word, the thing is easy. But if you don't understand the meaning of the word, thing will be like a rocket science. Whatever I am speaking you, it will just go above your head, just like that. Fine. So what is hypoosmolar? Hypo means less. Osmolar means osmolarity is less. When the osmolarity will be less? If the constituents are less in the urine, then only it will become hypoosmolar. If hyperosmolar, hyper means more, osmolarity more. Why osmolarity is more? Because the constituents are more. So if it is a hypoosmolar, the specific gravity will be less. If it is hyperosmolar, the specific gravity will be more. As simple as that. Just break the words to understand the terminologies. Okay, that's all. And thank you.